So it is my great pleasure today to present this year's Conklin Award awardee, share my slides here, to Scott Fraser. This is particularly special for me to introduce Scott. This award is, as I'll explain, is given to a outstanding developmental biologist and mentor, but particularly special for me because he was my postdoctoral mentor. By way of disclosure, the president has nothing to do with the selection of the Conklin awardee. That's all managed by the board and by the president-elect. And I recuse myself from all discussions, but I have to admit that I was quite pleased with the result. So let me take this opportunity to tell you a little bit about this award. The Edwin G. Conklin Medal was established in 1995 and is awarded annually by the Society to recognize a developmental biologist who has made and is continuing to make extraordinary research contributions to the field, and is an excellent mentor who has helped train the next generation of outstanding scientists. And you can see by this list of recipients, Scott joins an elite group of really phenomenal developmental biologists who have truly embodied the spirit and really moved our field forward. Edwin Conklin's work, I want to tell you a few things about that. He was a major figure in developmental biology working at the turn of the last century. He was a graduate student at Johns Hopkins and later a longstanding department chair at Princeton. And I should say there's a beautiful biography written and linked on the SDB website for you to refer to if you'd like to know more information. Like many of us, Edwin Conklin spent time at Woods Hole studying translucent embryos like marine snails and ascidians, and he utilized these pigment variations in the cytoplasm of these organisms to track cell lineage. And this really led to a beautiful analysis of the emergence of biological patterns and provided a clear enunciation of the idea of inheritance of cytoplasmic determinants in directing cell fate. He's one of the very few developmental biologists honored by Time magazine, probably the only one I could name. And so this is really a testament to the outstanding foundational work that he did and the importance of that. This honor is particularly fitting for Scott, and this is a picture of Scott at Woods Hole. I think this is around 1991. Scott spent many summers there teaching in the Woods Hole Marine Lab Embryology course. And he, like Conklin, has dedicated a large part of his career to studying complex cell lineages. In fact, Scott's work has brought numerous cutting-edge tools to the study of these complex lineages, and one nominator in his package wrote that Conklin would certainly use Scott's methods to trace lineages if he were alive today. Conklin also really argued for the importance of experimental manipulations to challenge and reveal developmental mechanics. And this has been a focus of Scott's career and a passion and forte of the Fraser Lab and many of the people who have emanated from it, even myself, for many years. So for more than 4 decades, Scott's worked to apply the tools of engineering, chemistry, engineering, and physics to problems in biology and medicine. He's a biophysicist, he's a microscopist, he's an engineer, a developmental biologist, a translational scientist. I probably could come up with a few more things to call him if I were pushed. He trained as an undergrad in physics at Harvey Mudd College, one of the Claremont colleges in California, very well known for their physics program, and was a grad student studying biophysics at Johns Hopkins University, which is coincidentally where Conklin was an undergrad. While he's advanced in his years, he didn't quite overlap with Conklin during that time. He directly joined the faculty at UC Irvine after finishing his graduate work, which is rare amongst our field, no postdoc, straight into a faculty position. And he joined the UC Irvine Department of Physiology and Biophysics. At UC Irvine, he really catalyzed the development, helped to participate in the development of the Developmental Biology Center there, an inter-school organized research unit directed then by Peter Bryant, and collaborated with many of the amazing labs there. And I know some of you are at UC Irvine, so shout out to you all. He became the deputy director of the DBC shortly thereafter. And during this time at UC Irvine, he started what ended up being a quarter century of service to the Society's journal, Developmental Biology, both as an associate editor and an editor. He moved to Caltech in 1990 to become the N.L. Rosen Professor of Biology and the Director of the Biological Imaging Center. And this is where I worked under Scott. Here is where he really began to cement his cross-disciplinary vision. There were chemists and biologists and physics all working side by side in really dynamic and fertile environment for innovation. Incredibly exciting candy store of science to work in, and a really great environment for many of us who spent time there. He helped to found the Kavli Institute of Nanoscience at Caltech, as well as serving as the founding director of both the Caltech Brain Imaging Center and the Rosen Center for Biological Engineering. And his innovations have spawned several startups and been integrated into instruments and FDA-approved diagnostics, working with companies like Zeiss and Leica to Genmark. In 2012, he moved over to the University of Southern California, and there he's had both a professorship role and a leadership role. As a provost professor in the Dornsife College of Letters, Arts, and Sciences and the Viterbi School of Engineering, with formal links to Children's Hospital of LA and the Keck School of Medicine. He really continues to push forward into disciplinary research, and it would be too lengthy of an introduction for me to talk about all of the amazing things that Scott's done, and I'm sure he'll touch on some of those during his talk, so I'll leave you, stay tuned for that, and he'll have the movies that accompany it, which will make it a lot more fun. He also serves as the Director of Science Initiatives at USC, so really a leadership role for the whole campus to utilize his innovative spirit. And he co-directs USC's Bridge Institute with its founding director, Ray Stevens. Scott's impact on trainees really is impressive. There's a long list of phenomenal people that have worked under Scott. Very proud to call these folks my brothers and sisters in science. Many of the names you'll recognize here are Developmental Biology Society members, and some even past presidents and current presidents and people who have served on the board, and really a phenomenal group of people dedicated to developmental biology through the time in his lab. And one of the things that I think is remarkable about Scott, he really provides this amazing environment for people to develop courage and really go into areas and think about areas that that aren't well formed. There's no path yet, and it really breeds a tremendous amount of courage and confidence in individuals. So they not only, I think, it not only breeds innovative work from his lab, but he really, many people who have gone through this lab have also taken leadership roles, building on the courage that they learned there. Scott is a consummate communicator. I think all of you, or many of you here, have heard him talk and gesticulate. He really, I think, invented the elevator pitch. He's always excited and happy to tell you about his work. Even if you're on a boat and trying to mind your own business, he's happy to come up to you and talk to you about the amazing things that he's done. Some of you may also know that he's also a devoted parent. His kids here were young, now they're grown-ups, and he's most happy when he's at the beach, where he will be talking to you from today. He's a consummate foodie. Many of you know that if you're at a meeting, you really want to get invited to dinner with Scott Fraser because he's picked the best restaurant in town. But to all of us who've trained with him, we know him as our favorite sweater vest wearing techno weenie, which has been memorialized on numerous occasions and fantastic Photoshop mock-ups shown here. You never know what Scott Fraser is going to do next, and I ask him from time to time, what's next in this amazing career that you've had? of all the people I know, I think he's the most likely to call me and tell me that he's going to follow in Bezos and Branson's footstep and tell me that he's headed off into space someday. And a few years ago, folks in his lab, I think, had a premonition, and so they helped to start outfitting him in his spacesuit. So I'll show this slide. With that, I'll introduce to you the 2021 Conklin Award medal recipient, Scott E. Frazier. So Scott, take it away.
Well, thank you very much, Mary. It's really nice to be the second speaker in this time slot. And that's, I'm glad that you were kinder than you could have been in the introduction. So, I promise to follow your example, and I guess mutual shared annihilation or something, whatever they used to call it with nuclear arms races, is something that probably keeps us both in our best behavior. Well, what I'd like to do today is to build a little bit on some of what Mary's talked about in thinking about Conklin and in thinking about the field, and also talking a little bit about the way that the field can be taught, as we heard about it from the previous speaker, in talking about how we are going to eavesdrop on developmental biology. And so why imaging matters is something I want to try to get across today. I want to start a little bit with mentioning a few things about Conklin, because in many ways he's a hero. He worked on cell lineages and cell fates. He was at Hopkins with an amazing group of classmates. So Ross G. Harrison, who was my scientific great-grandfather, was there then. Thomas Hunt Morgan, who many of you may know as a founding figure at Caltech's biology division. E.B. Wilson. It's just a phenomenal place. Trinkus was there a few years later, and Trinkus's comments to me at the end of the embryology course was, Hopkins is a great place to leave. And he was right in so many ways. He also took advantage of both the people and the resources, the experimental resources at the Marine Biological Lab. And I really owe my career to MBL. I mean, so as many people have talked about, you know, Conklin is the consummate person about talking about lineages, but he really lived for the environment. He worked at Woods Hole, he founded departments, he led departments, he was an amazing steward of the field. And as many others have said, like Phil Soriano and Rob Krumlauf and Richard Harland and others, SDB is really our favorite society. I mean, it represents a style of doing science and a style of interacting that I think we should all be very proud of. I love the journal, and as Mary mentioned, I've been involved with it for a number of years, and I'm very pleased to see how well it's doing and look forward to the next generation. And of course, Marine Biological Lab was my favorite lab. This is a historic picture from 1977 when I was a physics student trying to be dropped into the deep end of the embryology course. I came back the next year to do a little bit of TAing in the course, but then came back as a Grass Fellow in '79, came back as a faculty and then a co-director, and was a Forbes lecturer for the Grass Lab later on. And I guess the thing that MBL really got across to me is the importance of the style of interaction. It's got the world's best library, libraries where the papers end with all the things they wish they could have done, but the techniques weren't there. And that was a real inspiration for me of how to make things really work. The other inspirations there were two. So Kathy Tosney and Sally Hoskins and Tom Poole set up a discussion group and convinced J.P. Trinkus, who just had a new issue of Cells into Organs that had come out, to get together with us and have a little study group. And it was just amazing. Number one, it taught me the power of studying cells in the natural setting. It also taught me a no-holds-barred style of interaction. When Trink was trying to show me how to decorionate embryos, and I didn't do it perfectly for the first time, his encouraging comment to me was to ask if I had had a stroke. So he was not famous for nurturing comments, I guess. Somebody who was famous for nurturing comments was Ann Warner, who was there as a Lilly Fellow, and in the embryology course, she was working on a project with Tom Poole trying to study how cells were communicating by gap junctions, and it meant we were trying to impale cells as they were migrating and trying to inject dye into them that no sane person would have tried to do. And so Anne really got me going on this idea that we could label cells without thinking about what's impossible, really redefining what's impossible. And the other thing that MBL showed me was the importance of debate and argument and everything else as a path to understanding. And in 1978, I came back as a TA for a part of the course. Sue and Peter Bryant were faculty members that year, and they worked on limb patterning and limb regeneration and such. They invited Lewis Walpert and John Saunders to speak at the course that year. Both of them felt that what the Bryants were doing was wrong. Both of them felt that what each other was doing was wrong. So it ended up being this amazing week-long debate of Lewis Walpert versus John Saunders. The experiments that many of us know about grafting the zone of polarizing activity from the posterior to the anterior margin and getting a bifurcated limb, Walpert had formalized it by calling it the zone of polarizing activity. Saunders referred to the Walpert model as the zone of postulated activity. And so you can only imagine the sort of clearinghouse of arguments and experiments that came out of that. And it was fascinating seeing the next decade of work that was really set in that week of them arguing with one another almost nonstop. And also partying together and drinking together and eating together. So it was a friendly debate, but a no-holds-barred debate. In fact, MBL was famous for doing these sorts of things. The neurobiology series that was there on Monday nights is called the Monday Night Fights. And that was putting it mildly. So what I learned there was very much being dropped into the deep end of developmental biology. I learned to start looking for big questions that I didn't think were yet approachable, but could be. And one that fascinated me is something that Sidney Brenner had come up with, is this dichotomy about cell lineages and cell fates being guided by the American plan or the European plan. What he had thought about in this dichotomy is the American plan is sort of keeping up with the Joneses, or really letting your cell fate be dictated by your neighborhood or your neighbors. European plan, much more ancestry dictated. And so this is a fascinating sort of question. And of course, it was a fascinating question that had really motivated Conklin as well. And of course, the problem with much of the field is that reconstructing answers to that was being done from either fixed specimens or fixed models, like you can see this whole group of them in front of Conklin, and trying to deduce what might have happened. And of course, this is a horrible challenge, and it's a challenge that's always underspecified. And just to try to give you a feel for that, I'm going to repeat an analogy I've used before, which is this is like trying to understand a sporting event that you've never played, you've never seen before, and all you can see is dead players caught in snapshots. So if I was trying to understand the rules of American football, well, in fact, if I already knew the rules of American football, I could look at this and I could understand what's going on. I can see the ball carrier. I can see that he's about ready to be knocked over by another one of the players. But if I didn't know the rules, and if those players were all dead, and I looked at another game at another point, I wouldn't necessarily know what the rules were. I wouldn't even know what the important events are. In fact, the signal to noise is really hard in a football game because we've got things that are really football and things that are clearly not football all going on. So what's the signal and what's the noise? And I'd like to argue that even if we dropped individual NFL games into liquid nitrogen and freeze, arrested them at different points every minute or every second or every millisecond, it wouldn't help us as much as if we were using imaging. And so if we're using imaging, even these bad movies of bad games tell us a lot more about what goes on in a football game than dead players would. And so this is my dream experiment. It's the blimp above the Rose Bowl, all the players are there, everything's ready to go. And now the cruel joke about all the systems that I cared about is that tissues scatter light, so it's like filling the Rose Bowl with milk. And of course, the players don't come in uniform, so it's really nudist football. And so trying to make those cells distinct enough to be able to follow them and being able to see them as the challenge. Of course, that's where Conklin really had the whole thing down. And that's really captured in the Time magazine cover that Mary showed you earlier. This colored cytoplasm ends up being inherited unequally by the later cells as they're dividing. And as a result, he could look in and see which cells inherited it and know then which cells arose from which other cells, at least for some limited numbers of the divisions. And of course, they were nice transparent embryos, so he could see them over time. Now, in the systems that I care about, and you've heard about this before in previous lectures for Conklin awardees, like Krumlauf or Soriano, using transgenics to make the cells distinct. In my case, I used a more brute force technique, sort of based on what Ann Warner had taught me, and what Gerhardt and Gimlick and Keller and a number of others from the Leach community had taught me, using exogenous labels to make the cells distinct. And so this is a paper from years ago now, going into a chicken embryo and labeling two different groups of cells with two different colors, sealing it back up and coming back a couple days later. And of course, what you can see now is the cells that have inherited it. So the great grandchildren of these labeled cells. And we can reconstruct in the same way that Conklin could, the lineage from the intrinsic colors, we could reconstruct the lineages, or at least the patterns for this population of cells. Now, to try to carry this to single cell level and really ask questions about American plan versus European plan, we used a sharp micropipette, the sort that Ann Warner was using to label cells in the embryo, to inject cells after the injection to characterize where the cells were in a couple different views. You can see from the top view and the side view. And of course, the trick here was to keep them alive. And that's what Ann had started teaching me back in 1977 in the Woods Hole course. This is so alive that by the time I got to photograph it, the cell was in the middle of mitosis. In fact, it went on and divided and ended up contributing to the later development. We could take that same embryo and characterize where the labeled cells were, say at 10 hours, so we can see where it originated, where it was shortly after, and then score it after brain areas came about. And what you can see here then is that we can reconstruct a fate map. A fate map is what the cells become if they're unperturbed and everything else. So by taking the positions where they're labeled, and here they're color-coded at 6 hours and 10 hours, and labeling them by what they become. You can see there's a little bit of slop and overlap at the 6 hour time point, less slop by the 10 hour. And of course, this sort of effort is what really drove a lot of the work that we were doing in a variety of different systems. Now, Conklin is one of the people that I think really gave birth to experimental embryology as a field. He felt that observation wasn't enough. And in really learning his lesson, this shows what we can do to challenge a fate map and ask mechanistic questions. For example, if we take a cell and graft it up into the middle of that embryonic pattern that we were studying before, this at 6 hours, that would be grafting some of the cells that are destined to become muscle into the middle of where cells are destined to become forebrain. You can see that what happens is that a control graft gives you a normal animal, a normal forebrain, a normal eye, normal Crox-20 slices in the hindbrain. After the graft, you can see that this fate map is not a commitment map at all, because these mesodermal cells have now caused a duplication of structure. We now have 2 hindbrains in the animal, and now it's an animal that completely lacks forebrain. Very interestingly, this animal grew up to become a dean of research at a medical school later, because it lacked the forebrain. Anyway, so the sort of motivation for the early work was American plan versus European plan. And as with any of these dichotomies, of course, it's never going to be one or the other. In fact, the answer is always going to be both. And in this case, we got a whole variety of boths. Some were way more American, like the neural crest and in the retina and in the spinal cord. We found that second cousins and first cousins and brothers and sisters could be very, very diverse. It looked like very much like it was the American plan in operation. Others, like the hindbrain, looked much more European. We could see that by going in and labeling cells with the pipettes early or later and finding clones that ended up early on occasionally violating the segments in the later hindbrain. Later in development, we ended up with clones that never violated the hindbrain segments. And when we looked at the cell types, you can see in this lovely picture that Clark took that there's the clones end up being a whole group of identical, in this case, I think it was 16 sextuplets or whatever you would call it if you had 16 identical twins. So this is a cartoon from the paper that cover went with, just showing that the variety of clones, 8, 16, 32 cells that were non-randomly the same. So this is clearly looks much more European than American. And of course, we have to keep our minds open because we still have to challenge this. And in all of these, I think we get answers that are temporary answers. And these answers can change very, very quickly. In the mid-90s, when we were teaching at the Woods Hole Embryology course, C. elegans, when the lectures happened, was very, very European in its development. And Joel Rothman went away to the worm meeting. He came back a week later, and surprisingly, in 1 week, C. elegans had become American. The experiments had been done that showed that the local environment and the developmental constraints and the signals mattered tremendously. And so we have to take all of these answers with a grain of salt and keep on asking them over and over again. This is where Trink really helps, because what Trink just drilled into me starting in '77 was that we needed a way to follow the signals, the mechanics, the constraints. I don't think he'd ever use the words gene regulatory network, but I will. And he viewed imaging and imaging within the embryo, within its native setting, as the most critical for analyzing things. So, of course, that means that we've got to make ways to image it. That's where we've been trying to live, and that's where we've been trying to understand the developmental mechanisms and the developmental mechanics by living on this sort of pyramid of frustration. What we really want is to image the whole embryo at infinite resolution and super fast and everything else, but we have to realize that we always have to trade one off for the other. What I want to do is just take a few minutes to talk to you about how we can try to live happily on the pyramid of frustration by dealing with multiple labels, because to follow the multiple things we need to see more than one label at a time. The problem is any fluorescent dye overlaps. This is a problem that frustrated us and frustrates us to this day, because the spectra from different fluorescent proteins. In this case, there's 5 different fluorescent proteins and 3 different intrinsic signals that I'm plotting here. You can see that a normal way of taking a bandpass filter and trying to isolate one versus another is really not going to work well. And that's a technique and a problem that we've been working on for years, trying to use linear unmixing. And in fact, it's something that Mary Dickinson really pushed forward when she was in the lab. This is one of her historic images where each one of the spectral bands that you might take for cells labeled with fluorescein and with GFP overlapped one another. But if you take all of them and use the background spectrum that you know exists for the two, you can unmix the signals by taking a spectrum and then unmixing the data. The problem is that this is slow and noise propagates. And we were butting our head against this because it meant that the weakest signal always dominated the signal to noise. We were butting our head against it until Enrico Grattan and Francesco Cutrale and others told us how we were really doing this incorrectly. And what they were using was an approach called the Phasor, taking a region that would normally have a red spectrum, taking its Fourier transform, which becomes a single dot. A green spectrum becomes a green dot, a 50-50 mix becomes halfway in between. And this made the problem a forward problem instead of an inverse problem. It made it tremendously faster, in this case going from a couple hours to fully process 5 gigavoxels of data to tens of seconds. Francesco's now advanced it to where it's tens of milliseconds to process. This shows some of the results now of separating out these different signals and shows that we can live on this pyramid of frustration and be able to separate out some of the intrinsic signals, the autofluorescence, the structural signals, and the exogenous signals, in this case, fluorescent proteins, even though their spectra overlap one another tremendously. And he also, with his collaborators, Francesco has just been doing an amazing thing, has been dealing with one of the things that's been limiting us tremendously, because we want to be able to image with very few photons. We want to make sure we're not damaging the specimen at all. And as there's fewer and fewer photons, instead of a beautiful spectrum that would happen with thousands of photons, once we get down to dozens of photons, the counting error just gets to be horrible. Luckily, Francesco and his colleagues were able to do a deep learning approach. That deep learning approach allowed them to reconstruct the spectrum, and it allows us to take data that we have no business trying to interpret and put it through their new algorithms and be able to see now the cell boundaries and see multiple labels and see it with maybe 10 or 20-fold lower photons, which is really allowing us to image things with much better precision, much higher spatial and temporal resolution, and even look at things that we could have, where we could have perturbed the embryo. Now, of course, since this technique got such great sort of signal to noise, it can also see the background signals. And it can separate those background signals into things like the retinoids, like retinoic acid, NADH free and bound, which is a way of seeing what the metabolism is of the cells. So, of course, this is exciting for me because we eventually want to apply this to people. And of course, there's very, very few people, except for maybe some of us experimentalists who have been working with GFP viruses who have human transgenes. On the web, I was able to find something that must only have been Lewis Wolpert's dream. It's a developing human with a French flag on it. For those of you that know the Lewis Wolpert French flag model, you'll recognize why that might be important. But anyway, there's very few of these around, and I'm not sure I could afford the colony. But what we could do is to go in and use our ability to see these autofluorescent signals, as well as to be able to see the exogenous labels, to see some of the things that we know matter, like retinoic acid as a signal, or to be able to see NADH bound and free, which allows us to see the metabolism of the cells. Now, this works well with color. It turns out it doesn't work as well with color as we go to bigger specimens that scatter light more. And that means that the way that we really could answer this is to use different technologies, not look at the color of the fluorescence, but the lifetime of the fluorescence. And it turns out that the Phasor technique works lovely there, but that's a whole nother thing for us to talk about. It's a topic for another day. So what I'm going to do is end there and just sort of point out that we have the ability now to imagine looking at exogenous and endogenous signals to ask questions that Conklin would have loved to have asked, but couldn't. And the questions now that we need to ask about are the things that we as a field care about. And I guess I should end just by pointing out the unbelievable support I've gotten from my colleagues. I mean, I'll just use one example of this. When Mary was in the lab, I was on my way to a cell biology meeting to give a talk. She was doing this amazing stuff at the time in the mouse embryo of looking at how it is that the shear forces and other cues were shaping the cardiovascular system of the mouse. I thought this would be a great time to really run Mary's work up the flagpole, so I asked her for slides. Mary, of course, complied, and this is the first slide. And for those that you didn't notice, many of the key features here, apparently my name is not Scott Evil Phasor, but Scott Egghead Phasor, and the Scott has been dropped. And my email address has been changed. And of course, she used the font that the people in the lab always scolded us for using, Comic Sans. So anyway, I have had the luck over the years of working at Woods Hole and a variety of other places with people that have really shaped everything that we've been able to do. From the work we did in the biophysics department, first at Johns Hopkins, earlier at Harvey Mudd College, at UC Irvine, Mu-Ming Pu was an amazing collaborator. Hans Meinhardt came and wrote his book while living at our house, which was just great fun. The mouse and mouse course community was amazing for introducing me to problems and also to many solutions. Liz and Rosa both stressed on me the importance of rolling the lime before you cut it to make your gin and tonics. I think they just impressed that on me so that I would slice the limes for their gin and tonics at the Cold Spring Harbor course. But we, over the years, were able to work on Hydra, sea urchin, develop MRI technologies, fly, avians. It's just been an amazing course. And I just really appreciate everything that everybody's done in shaping what we could do. And I also appreciate the amazing group of colleagues I have right now and funding agencies that have given us the freedom to do things as crazy as what I've been telling you about today. So I'm going to stop there. Thank you for your attention and thank the Society for this amazing award, which just makes me very happy given the origin of this whole project.